From Microbe TV, this is TWiP, This Week in Parasitism, episode 111, recorded on June 2nd, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code Microbe. Joining me today here in New York City are Dickson Despommier.
Hello, Vincent.
And Daniel Griffin.
Hello, everybody.
And a penguin.
Hello, Daniel.
And we have a penguin here also.
We have a penguin, we have an imaginary penguin.
Penguins are really short, aren't they?
Well, not all of them.
Are they?
The
some
emperor penguins are quite tall.
Are they tall as humans?
No.
We didn't have any emperor penguins at the Bronx Zoo, did we?
No.
We had Magellanic penguins.
Yes, those are temperate zone.
I must say I learned a lot, and I did not realize that birds carry, like sparrows, carry malaria around.
Come on, that's how Ronald Ross discovered it.
So in my backyard we have feeders, and this morning there were two goldfinches.
Nice.
Which you know is the New Jersey state bird, I bet.
I didn't know that.
Do you think they have malaria?
Oh, they could.
I would say most likely.
They could.
Can I get one and squeeze it up and see?
If you trapped it, it would probably be a federal offense. Because
think?
It's the state bird, don't you remember? You know what the state fish is? Believe it or not, it's
trout?
brook trout. It's the brook trout,
which
almost doesn't exist in New Jersey anymore.
If I caught a goldfinch, then Chris Christie would come running after me, huh?
Somebody would.
And you know what? You could probably outrun him.
I bet I could.
Right.
I
know someone who, when I was at Rockefeller, Curtis Patton, had a student from high school come into his lab and say, I want to work with you for the summer. And Curtis said, sure. What do you want to work on? He says, I want to work on rat parasites. He says, okay, let's go to the New York City subway and trap some and see what they have. And they did. They had some interesting things.
Ian Lipkin has trapped rats and found things here in New York City.
Yep.
All right. So we're back in our usual place. There's no exotic locale today, right?
Nope. Well, I don't know. Looks pretty exotic out the window right now.
What is it doing out there?
It's hazy, slightly overcast, a few scattered clouds, I would say. Not overcast altogether, but it's starting to show signs of humid summer-like approaches to the atmosphere, accumulations of pollution.
23 Celsius and sunny. Tomorrow's
the humidity.
I don't know. It doesn't say. It's going to rain tomorrow.
Is it? Oh, really?
We had a case last time, Daniel.
We did.
Let's summarize it. We had lots of guesses. For the penguin, we didn't have many, but people seem to like having human cases. We have lots of guesses. Why don't you remind us what we're dealing with here?
Definitely. Now, as I mentioned last time, this week's case involves humans, actually, several humans. A young woman in her 40s who was concerned about bug bites. And the story we had gotten is that several weeks ago, her 13-year-old daughter had woken up in the morning reporting bug bites. These were described as several small, red, raised, itchy areas. And there were 3 to 4 of them in a line. They're just above the belt line. And then we get a story. They leave. They go on vacation to Europe. There's no problems while they're in Europe. But when they come back, it's now several weeks later, the mom wakes up and she's got a similar 3 to 4 red, raised, itchy areas in a linear pattern just above her belt line on the lower abdomen. And then the second daughter has the problem. And then I should bring this right up because this was Dickson seemed very focused on. So the dad doesn't report any problems. So
totally allergic to the situation.
Yes, he's not reacting.
He's
not reacting to the situation.
The dad is living in the same house.
The dad's living in the same house, apparently sleeping in the same bed. We clarified that. We did give the little hint that the family lives in the New York metropolitan area, which is an epicenter for this problem. And these are always in the trunk. They're never reported on the arms or legs. And there was no travel immediately before the first daughter's bug bites. And the family does spend a lot of time outdoors. They live in like a suburban wooded area.
So I'm curious as to why Daniel always refers to them as bug bites. They could be from some other things.
I was going to ask you, what is a bug?
There is a definition for a true bug. It's an hemipteran. That is the order that they're in, the hemipterans.
But
so the reduvid bug.
Yeah, those are true bugs. Yeah.
Yeah.
Is a spider a bug?
Of course not.
It's
not even an insect.
Is a mosquito a bug?
No.
A spider is an arachnid, right?
That's right.
Is a mosquito an insect?
Yes.
Everything is an insect except mosquitoes? Except spiders, excuse me?
No, anything with 8 legs is an arachnid. Anything with 6 legs is usually an insect, but some nymphal forms of arachnids can have 6 legs.
But people use the word bug for everything.
They do, but in this case it was...
How many legs do ticks have?
I think that...
How many what?
How many legs do ticks have?
Ticks have 8.
But that's, I think, what you bring up. At some nymphal forms, they can have 6. So you...
The developmental stage leading to the adult...
So we don't call them arachnids?
Sure we do.
Even though they have 6?
Well, because they're larval, they're nymphal.
Yeah, but the adult stage is what's classifying them.
That is the one.
All right, so spiders, ticks, what else have 8 legs? Mites. They might have 8 legs.
Spiders, ticks, and mites. Those are the best ones to keep in mind. And mites...
How did you make that symbol with your hand?
Well, you go like...
I have a movie of you doing that on TWiP. All right.
So, when you say bug bites, I would say...
You want to say
you were
arachnid?
bitten by something.
Well,
something.
Yeah, that's a good point.
Our job is to find out what.
Why did you say bug bite? I'm just curious.
Oh, no, that was actually the chief complaint of the
woman.
I have what I think are bug bites. I'm concerned about these bug bites.
You can censor that.
And I, of course, spent several minutes. Well, you realize that when you say bug bites, you may be referring to an arachnid, an insect, or a true bug. No.
Okay.
You did say that to her?
He goes like this or like this.
Did you really go through all that with her?
No.
Yeah,
But this would, if you were my child and you said you had a bug bite, I might torture you with things like that. But they would.
Well, they would remember that though.
They would actually.
Do true bugs bite?
Absolutely. No, reduvid bugs are bugs.
Reduvid's good.
Sure, and there are other bugs which we may or may not.
Are beetles bugs?
No, they're not.
They have 6 legs though, right?
Beetles are coleopterans.
And
you want to quiz me on all the orders of insects?
No, I'm not quizzing you.
It was on my preliminary examination when I was a graduate student.
I'm just curious. I like hearing about classification. It's very interesting. Systematics I always liked.
Sure.
I wish I could remember it all, though.
Well, I couldn't either. I forgot Coleoptera, of all things, on the...
Got it. All right, we have a lot of letters. Let's get through them. The first is from Michael. Hello, gentlemen. After listening to Dickson becoming highly suspicious of the fact that the husband did not have any bites, and I listened to this over and over many times, even at half speed.
Yes.
I am convinced that said husband would get up in the middle of the night and scrape mite-laden pigeon poop off of the windowsill and sprinkle it on the exposed midriffs of the unsuspecting victims as they slept.
Not fair.
Or maybe the women were leaning out over the encrusted windowsill and waving to the neighbors during the day while still wearing their pajamas. Wait, isn't New York City the bedbug capital of the world? Nah, that would be too easy. I'm sticking with pigeon mites. Mike in Oregon, so he thinks pigeon mites.
That's a good guess.
Okay.
It's a good guess. But another thing I was driving at with regards to the male of the family was the fact that if he's a middle-aged person, he's usually up 3 or 4 times at night peeing. And this may not allow enough time for whatever causing the problem to attack him.
So there is something good about aging.
Only one. That's it.
So all the older men that listen, when you get up in the middle of the night multiple times, you go, "Oh, there is some protection there,
some redeeming value to that."
Right, Daniel, take the next.
Jan writes, "Gentlemen, again, we're all being referred to as gentlemen. I like this.
Love it.
After a brief break from sending in guesses, I thought to give this one a try. Bedbugs." Itchy, small, red welts in a regular pattern after waking up and living in the New York metropolitan area. Are there any other candidates? As for the husband not showing any symptoms, Dickson's suggestion of separate bedrooms makes sense, especially if his bedroom is way off from the others. Or, for instance, he might be a really loud snorer. Greetings from overcast, drizzly Rotterdam, temperature 16°C. So do you think the really loud snore is keeping things from biting him or keeping him from sleeping in the same bed?
It's keeping him from sleeping in the same bed.
All right. So, Dickson, here you
All
go.
right. I'm going to pronounce this two different ways so as not to offend. Tubi, or Tubby, writes, Greetings, TWiP trio. I usually don't venture any actual diagnosis for your cases because while I find listening about parasites to be interesting, I find the reality of them to be horrifying. So I decide every episode that it's malaria, and sometimes I'm even right. Like last time, for instance. This time, though it sounds like a case of not so squicky? Not so squicky bedbugs. Never heard that word squicky before. Cymex lectularius. Ectoparasites are parasites, too. That the husband is unaffected is interesting, and I look forward to hearing an explanation as to why that might be. It would be nice to know if the younger daughter had a sleepover at a friend's house, or if she stayed overnight in a hotel in Manhattan shortly before the symptoms appeared at home. While living in New York, I was advised to keep my mattress in a waterproof vinyl bag so that I might not have to replace it if I moved into an apartment where there were already presence of, or I brought them home from a movie theater.
Yuck.
Yeah, imagine sitting in a movie theater and bringing home bed bugs. That's a bad thought. I mean, I didn't say it's incorrect. I just said it's an awful thought. Too bad.
Wink. Next one's from Wink. Dr. Wink writes, Dear professors, I'm getting so many TWiP cases wrong that I have sadly concluded that I have the wrong skill set. And this is from an infectious disease physician. Oh, boy. This week I asked my wife for guidance. Lynn is an artist and a designer, and she thinks the ladies are being bit by mites. She wants a skin biopsy.
Interesting.
Mites. So
it's not out of the question.
We have 2 mites and a bed bug so far, huh?
We do.
Okay. Dave writes, Dear TWiP trio, after 2 weeks of not emailing diagnosis and extremely baffled by the case of the penguin, I finally feel confident enough in this week's case study to venture a guess. I will say that the 3 afflicted family members were dined upon by bed bugs in New York, the 13-year-old daughter, and Europe, the mother and older daughter. The reasons for this guess are because of the show notes mentioned. New York is an epicenter for this problem, and everyone knows bed bugs are not uncommon in the New York metropolitan area. There are, however, also common in many European countries, and the fact that almost every member of the family was affected is merely coincidence. Bed bugs tend to gravitate to warm areas on the body, the trunk, and their bites appear in line form as itchy, raised rashes. When I last emailed, I had mentioned my acceptance into a PhD program at the Tufts Vet School, and you inquired as to the name of the PI. I will be working under Dr. Patrick Skelly, studying schistosome tegument proteins and host-parasite interaction. Thank you again for the informative podcast. Sincerely,
nice.
Dave B.
Nice.
See,
you like that?
I do. I love that. In fact, I do. I like that a lot.
Schistosome tegument proteins.
Like it.
The next one.
Yep.
Let me pass this over. This is from Elise. Elise, you're back.
Ha. Elise writes, Dear TWiP trifecta, I hope this finds you well. I also dearly hope you don't get any pushback from listeners about the misleading aspects of last week's case study. I would never mind hearing about any non-human cases, and I was happy to hear about the penguin patient. It was such a tremendous pleasure to hear Paul Calais' case study and hear about his work, at the Bronx Zoo, which I love visiting, and I'm hoping to be there over the Memorial Day weekend. I also loved the video this week, not only for getting to see all of you, but also for getting to see the patient's environment. Since I truly have no background in sciences, there was no easy way I was going to be able to diagnose TWiP 109 correctly, and I didn't send in my guess, which actually was probably going to be Babesia, except I wasn't quite satisfied with the way all the symptoms presented in the timeline, and then I couldn't find another likely solution. Thank you so much for having Paul on TWiP. I am not at all certain about my diagnosis for the 3 women in the greater New York metropolitan area, but I suspect that the relapsing patterns of red lesions have been caused by bed bugs. Perhaps because I am in Lower Manhattan and in the city, where bed bug stories are legion, I am not quite convinced that I am correct because it seems a little too obvious. The clues that make me think bed bugs are the culprits are very basic. The 3 patients contracted these relapsing lesions consecutively. not all at once, which suggests that the parasites started in the 13-year-old's bed and found a way to migrate into the other two beds. As Dr. Despommier points out, the lesions show up in the morning, not after having been present the night before. The pattern of lesions fits the bedbug profile for taking meals in a row: breakfast, lunch, dinner, and a snack. The patients have no symptoms beyond the rows of itchy red bumps, which is standard for bedbug bite sufferers who don't have a pronounced allergy. No one experienced symptoms of any kind while the family was on vacation, and then the lesions began reappearing after everybody got home, where the bedbugs, if I am correct, were house-sitting for them. As to how the bedbugs could have made it into the house, there are countless possibilities. They could have hitched a ride in the gym or gear bag of the daughters, particularly if they are in the trunk of cars with lots of other kids' bags from a lot of other houses. If one of the daughters spent the night at a friend's house or took a school trip and stayed at a hotel, the bedbugs could have come home with her. There is one family member who has not yet developed any kind of lesions. I wonder if he had simply not noticed that he had been bitten. Some people are not sensitive and require a greater number of exposures over a longer period of time to start reacting. Another possibility is that his wife is more appealing to bedbugs. I am one of those people who gets bitten by mosquitoes when no one else around me gets bitten, so perhaps it's possible for something similar to be happening. I actually somewhat hope I am mistaken in this diagnosis, because getting rid of bedbugs is a huge pain. Thank you so much again for the wonderful case of the unnamed penguin. I am glad he recovered. And thank you so much for the TWiP. It's a pleasure.
Brendan writes, sounds like bedbugs. Regards, Brendan.
Oh, I could have read that one.
Gregory writes, hello, fellow TWiPers. I am a recent undergrad graduate from St. Joseph's University. Regarding the case study this week, the signs of this parasite sound familiar to a common college dorm infection, scabies. My thoughts are that the red lesions, particularly around the waistline and legs, are in dark places and folds of skin, common locations for scabies rashes. This explains why it would spread from the daughters to the mother. However, I'm not sure why the father has not contracted the lesions. As for why the rash subsided when they traveled, my guess is that once they traveled away from home and subsequently their sheets, beds, couches, etc., the scabies were no longer in continuous contact. Thank you for your consideration. I am a new listener to TWiP, and I listen to the whole family of podcasts. I thank you for all your hard work, and I hope to learn more from continuous listening. Gregory.
May I add, Gregory, that this is not hard work. This is a labor of love.
You mean you don't work hard at it?
Nope. You don't have to work hard when you enjoy something.
That's very sad.
No, don't be sad, Vincent, please.
I'm really sad. Daniel, can you take the next?
Sure. Dear TWiPers, after last week's curveball involving a non-human host, it looks like you decided to pitch this one straight down the middle. So after taking no notes or doing any research, I am swinging for the fence and submitting Cimex lectularius as the culprit. You like that pronunciation? Cimex? It really should be Cimex, right?
Perhaps.
Shouldn't it?
Perhaps.
But anyway.
I've heard it Cimex only.
Okay. All this assumes that you did not become even more diabolical this week. I will note that I've read many accounts of New York bedbugs in the past few years, that I have observed that many times the bites are linear, and that the husband not being symptomatic only indicates that he is one of this significantly large percentage of people who do not react to being bitten. Never miss any of the episodes of any of Dr. Racaniello's podcasts. An impressive body of work. Thank you all. It is a perfect Greenville, South Carolina weather. Currently 17C without a cloud in the sky and a promise of warming to 28C later. Life is good. Don. All right.
I guess he has a coffee mug that says life is good on it.
I like those mugs and shirts.
He has LG television set, maybe. Jeff writes, hello, Trip Triumvirate. I didn't hazard a guess last time, as I couldn't come up with a parasite which would result in pulmonary edema in a 4-year-old with a history of raw fish consumption. Now I know why. This week's case is likely due to bedbugs. A little known fact about these pests is that not everyone will have a reaction to the bite. It is likely that the husband was bitten as well, but just didn't have the reaction that the wife and daughters exhibited. Jeff.
All right, the next one is from Jen, who writes, hi, TWiP team. Thanks for covering avian malaria. What a tricky case. Relevant to your last episode, I was saddened to hear about this recent report of loon malaria from Audubon.com. It appears parasites were also found in the brain of this bird, so could it have been acting more loony than usual? More seriously, what are your thoughts on this? Could an average bird watcher enthusiast notice a behavioral difference in an infected wild bird? And do you think this is an isolated case or a potential epidemic? My guess for the current case is bed bugs. I don't know anything about it. We should send this to Paul. Whether loons. Pathologists documented the first death of a loon in New England from avian malaria.
That's interesting.
As Paul said, some species get sick and others do not, right?
That's right.
Should we ask Paul about loon malaria? I should.
We could ask Bob Gwads, too. I think we should probably send out an all-points bulletin. Because Bob is a pretty good expert at different types of malaria.
All right, I'll forward that on to... I'm sorry, Jan, that I should have done this before and had the answer ready for you. I didn't work hard enough on this episode. I'm sorry.
I'm hopefully going to see Paul next Thursday night at the Central Park Zoo.
What are you going to do there? A fundraiser?
It's a fundraiser. We don't raise fun
or fundraiser.
Fun. There's no D for me.
Right. Exactly.
I'm there for the fun.
Next one, Daniel.
Heather writes, "Hello, TWiP. I think the ladies with the bites have a bed bug infestation.
Nice.
Possibly acquired by bringing home soccer uniforms or drama costumes that were in an infested home. For treatment, I suggest they burn their home to the ground with all their possessions inside and start afresh far away from New York City."
What a great solution.
As for why the husband is not reporting bites, what, if anything, do all four family members wear while sleeping?
Who knows?
The choice may be facilitating bites on the women. Or is the husband perhaps very hirsute? This may be TMI.
Is he wearing hirsute?
But since I stopped shaving my legs, I experience fewer mosquito bites. Okay. I'm not that hairy, but I think either the hair dissuades mosquitoes from landing or I feel them more quickly and swat them. I know two vets who have interesting elasma branch parasite case studies. One involves a professional sports team. I will pass along their emails if you want to try to recruit them to TWiP. Heather.
You've already given away the answer, right?
I know. That.
We didn't say which elasma branch.
Are there a lot?
There are.
All right.
Did you ask them if they slept naked?
I did not.
Not you, Daniel.
I do know the answer to that, so we'll bring that up. We'll discuss whether or not that's relevant.
Cool. If the guy does, I don't want to know about it.
Corey writes, Vincent/Dickson/Daniel. This week's case sounds like the classic symptoms of bed bugs. There's no need for a differential diagnosis. This is the breakfast, lunch, and dinner, which is often a euphemism for the set of linear bites associated with bed bugs. It is odd that the husband doesn't have any of the bites on himself, but they probably prefer the odor of others in the house. I know my wife will attract mosquitoes more than I will at times. Perhaps bed bugs are similar in their host homing behaviors. P.S. You may want to update the description of TWiP on your webpage. It still says Dick Despommier, and I know a while back you mentioned that he prefers to go by Dickson. All the best, Corey. No, Vincent keeps it there on purpose because everybody writes, "Be nice to Dickson," and he just can't seem to do it.
I'm not going to change it. If you hate it, I'm going to leave it.
No, I don't really care. I will modify this. If you call me Dick or Dickson, that's fine.
Don't call me Vince.
That's fine. Don't call me Vince. No, I won't call you Vince. I promise I won't do that.
Really?
Vince, I wouldn't lie to you.
Chris writes, I'm writing from Somerville, Massachusetts, and spring has finally arrived. 72F, the sun has set, and it was a really pleasant day. When I picked my first case to diagnose a few weeks ago, I initially thought I had an easy one, 107. I was proven a bit wrong. This time I had an idea of a diagnosis. Even while listening to the case history, the location was highlighted as important, and I know bed bugs are an issue in New York. It sounds like these skin irritations were appearing during sleep, so that's another check mark for bed bugs. CDC fact page on them even mentions the bites can appear in a line. It's possible the husband is getting bitten too, but his skin doesn't react. That's what I'm going with, bed bugs. I wish you all a good holiday weekend. Chris from Massachusetts.
All right. Mark writes, hello, TWiP trio. There is a definite source of infection. It could be the bedroom, but it could also be outside a window. Scabies would be consistent with the lined up linear series of bites. However, They would also be in the intertriginous areas, such as the webbing of the fingers. They wouldn't go away during a trip to Europe. Bed bugs would be a very generalized series of bites that wouldn't be restricted. They occur in lines, in clusters, in unclothed regions, and are not limited to one area such as the waist, which is probably clothed. Fleas don't leave linear patterns but clusters. They would bite where clothing is loose, such as waist, but also the legs. I am very stumped, but a guess would be bird mites. It is a nocturnal feeder and can bite where the clothing fits tightly, such as where elastic bands are located, though it can be generalized to other areas. Don't know, but look forward to the answer. 29 degrees C in Oklahoma today and humid. Okay, Mark writes.
Mark writes. Hello, TWiM DDV.
Team
DDV.
Oh, sorry. Hello, Team DDV. What did I say?
TWiM.
I'll help pronounce the little words. You can pronounce the big ones.
Okay,
there.
fine. Okay, fine. First, I want to congratulate you all and say well played with the subterfuge in TWiP 109's case discussion of the 4-year-old female. The reveal in episode 110 by Dr. Paul Calais that the patient was a 4-year-old.
Year.
It says your right.
I know,
here.
but can't you fix it on the fly?
You asked me to read every word. I would. Shocking reminder that TWiP was conceived to cover eukaryotic parasites, not just those of us Homo sapiens. I'm making an educated guess of which parasite for TWiP 110 is. There are not enough facts to support a good differential diagnosis. I nominate common bedbugs, Cymex lectularius, as the parasite for this show. Here are my reasons. Waking in the morning and discovering 3 to 4 small red collinear and potentially itchy sores is consistent with being bitten by a bedbug. See this note at the CDC's website, and then he gives a reference, a link. 2, the mother has been described as being a frequent traveler. Bedbugs infest hotels throughout the world. Bedbugs are commonly spread by hiding.
In the seams.
By hiding in the seams of luggage. Her frequent travel provides an opportunity for contamination. 3, the mother and 2 daughters are described as very active in youth soccer and bringing other girls into the game. This is also another potential source for contamination. Daniel describes the family as having a recent trip to New York implying Manhattan. He was silent on if it was a day trip or an overnight travel. Why is this important? Manhattan hotels have, in recent years, problems with bedbug outbreaks. Search Google for the New York Times article for more information. If they stayed at a hotel, then it's another source for contamination. Or if they checked bags or clothes at a theater or restaurant, there is another potential source for transmitting bedbugs back to their home. The metadata that Daniel communicated is equally important to me. That the scarce clinical facts. Yes. He commented that the collinear wounds are, I forget the exact word, either typical or classic for this parasite. Later, he commented that New York is the epicenter for this show's case. Case. See, I didn't say cause. I said
Very
case.
Good. You're getting really good.
Well, I just have to edit as I go. Responding to questions from Vincent and or Dickson about inspecting the home bed sheets, Daniel avoided answering the question. I seem to recall this is where he introduced the image of Dr. House, sending his staff to break into problematic case homes in search of environmental information affecting diagnosis. Too bad Vincent or Dickson didn't vigorously pursue their question. The CDC note indicates finding bedbugs stool, shed outer skin, or blood stains from the bites is important for the diagnosis. Thank you, Mark.
Eric writes, Dear TWiP Triumvirate, thank you for another fun case of the week. My guess is as follows. The mother and daughter are all quite gregarious, yet the father, unaffected, suspects the nefarious. Why do the ladies get bumps from their beds? All over their bodies, but not on their heads? He sleeps on the couch, suspecting Cymex lectularius. A nice little, what do you call it?
It's not a limerick, it's
Limerick.
a poem. It's a little
Limerick.
It's a nice little.
Seattle is enjoying sunny and 26C, a true delight in May.
AJ writes, Hello, TWiP team. I thought I'd take a break from my post-back applications to catch up on my podcast. And to my delight, there is a case I think I actually know. I'm writing in about the case where the humans have bug bites. I put those in quotes because it seems people blame bug and spider bites for a wide variety of skin conditions. Yeah, I think you mentioned that, right? We just have is that a red-tailed hawk about 10 feet from our window flying by?
It's a beautiful.
Is it a red-tailed hawk?
It's a red-tail. Yeah, it's a red-tail.
Sadly, I recognize this condition, and almost instantly, itchy bumps in a row of 3 or 4 appeared in the morning. No. Sadly, I recognize this condition almost instantly. Itchy bumps in a row of 3 or 4 appearing in the morning spells bed bugs. The pattern of 3 bumps is almost referred to as breakfast, lunch, and dinner. We had a prior person mention that. 3 or 4 usually. Right. This scourge is in San Diego, and I am unfortunately intimately aware of it. To clinch the diagnosis, yell at the patient, break into their house, and check the mattresses, according to Dr. House. Dickson seemed to be surprised that the male didn't have bumps, but most people don't actually get itchy bumps, so that isn't so surprising. I didn't even realize that I had bed bugs until I saw one. If you can't find the bugs, look for their fecal stains along the seams of the mattress, like someone dotted it with a sharpener or a Sharpie. Fleas would be my second pick for ectoparasites because of the linear bite patterns. I don't know so much about the dermal conditions, and although it possibly could be cutaneous larva migrans, but that doesn't fit so well, especially with the travel history. If it's bed bugs, I am very, very sorry, as they're extremely difficult to get rid of. Of course, the damage is greater psychologically than physically. Thanks for the inspiring podcast, as always. AJ, parasitologist wannabe, San Diego. All right.
Where June gloom is thankfully kicking in. Now, what does June gloom mean? Do you know?
In San Diego? I didn't think they got any change.
June gloom.
It might be the doldrums of hot, humid weather.
Dickson, in terms of here in New York City, the kinds of insects and arachnids that might, notice I say insects and arachnids, that might bite people.
You could say arthropods, and that would cover both.
Would that cover them all? Arthropods?
Arthropods.
We would have mosquitoes, fleas, ticks, mites, bed bugs. What else?
Lice. No one mentioned lice, but I expected to hear someone say,
like
could be body lice.
Like hair lice.
Head lice usually stay put on your head, but body lice.
Pubic lice, right?
Pubic lice. That's true. So no one mentioned them.
Is that it? 6? So there are 6 different kinds of arthropods.
At least.
Arthropods?
Yep. Yeah. Jointed legs. That's what that means. Arthropod.
Dixon crooks his index finger.
He did. As an insect.
All right. It's always good to know generalities.
Well,
do we need any more information for this case, Daniel?
I sure didn't.
Did they sleep naked?
What?
Yeah. So let's let you guys ask a few questions. So the husband sleeps in like long, basically pajamas, like a t-shirt and long pants.
And long pants.
And long pants, like those sleeping pajama pants. The mother sleeps in just a supportive sleeping bra and panties.
She's sleeping separately, right?
No, they're actually sharing the same bed.
Oh, okay.
The 2 girls, one is sleeping in a nightgown. That's actually the first one that gets bitten, and then the second has like a 2-piece pajama thing that they wear.
Yeah.
Yeah, it doesn't help very much.
Well, we forgot to mention one other biting potential, and that's chiggers.
In New York City?
Yeah, sure. The golf courses particularly.
Yeah.
And there are golf courses in New York City, by the way. So chiggers was a remote possibility only.
But that's a southern thing, too.
No. It's all over the place.
What is a chigger?
Chigger is a mite.
Mite. They go under your skin, don't they?
They do, but they usually bite around where the clothes are fitting tight.
Is that right?
Yeah.
They usually bite around where the clothes are fitting tight.
But never during the day, only at night. No, that's not true.
You're doing so well there.
No, they only bite during the day, actually. That's when they bite.
Nice. Okay.
Usually the reaction to a chigger bite is after the chigger is dropped off your body, so there's no way of finding the culprit. And that's true for these things.
You're such a font of information.
Well, I had several.
You should share it. You should share it.
Well, I keep it under my sombrero.
Yeah, actually, that would be. Why don't you do a podcast and then you could disseminate it for free?
No, we could never.
People would listen. I think people would tune in.
Some might. Some might.
Some might.
Yeah. Well, I hope you were catching on to that.
Yeah.
That's right.
So
otherwise, if they didn't listen, they would flee.
Oh, that was so good.
Hey,
so
Alan
Vincent
Dove.
wanted to know if people slept naked.
We got that.
So we got that.
You did?
What, do you have any questions there,
Sex,
Dickson?
politics,
Do you feel like you've got it covered? Do you feel like you know what this is already?
Did they ever try to trap any of these things and bring them in?
So you're going with the Dr. House suggestion. So.
Yeah, well, I would like to see evidence of the culprit.
All right, and so I will no longer evade. I'm going to give you that answer, but I wanted to sort of give some feedback to our writers, people who wrote in. So a couple things people talked about was the characteristic pattern.
Yes.
Mm-hmm.
And we're going to learn that characteristic pattern is going to fit with the confirmed diagnosis.
Correct.
Because people were bringing up the fact that there's possible different ectoparasites. Could this be fleas? Could it be bed bugs? Could it be scabies? So let's discuss the difference. So Dickson, you want to jump in?
Well,
I saw you.
No. I will
didn't
add
mention-
if necessary.
Yes, you're going to add.
You didn't mention scabies in your list of
No,
arthropods.
because
It's not an arthropod?
it's an arthropod. Of course it is. And it's a mite, but it doesn't bite.
Yeah, so
It's not bite.
the scabies are going to they're going to burrow in, and so they're kind of a classic. Well, as far as localization, we'll start with that. They tend to be in the webs between the fingers, between the toes.
Right.
The idea that they're going to burrow in sort of maybe an obese person in the folds. I've never seen that. So
that's not true at all.
You might see what we call a Norwegian scabies, which is this massive infestation of scabies. Then you start seeing things like that. But any sort of a mild infestation of scabies or...
We might want to add that the Norwegians are not particularly fond of having it called that any more than the Spanish are fond of having it called Spanish flu. So Norwegian scabies was unfortunately named after a gentleman who was immunosuppressed, and he happened to be from Norway, and his body was just covered with
That was actually
scabies.
described in Norway, right? It was in Norway where they did it.
Do they have a problem with Norway rats?
Norwegian rats.
You'd have to ask the Norwegians.
Weren't you bitten by one
Yeah.
as a child? That explains everything.
That never leaves your mind once you have.
Every
that
time
happen.
you react badly, I think of
Oh, really? You
bite.
think of that?
Your
Right.
bite.
You think?
No.
Now, were you harassing the rat? Were you like pulling its tail or something? Or
It
it's
did
just
worse
out
than
of
that.
the blue?
No, I picked it up by the tail, and
Yeah, of
the rat
course.
reflexed back on my finger and just bit and didn't let go.
I would bite you if you picked me up by my tail. Come on.
You have a tail?
We're not going there.
Some people have tails.
We're degenerate.
And then they sort of mess. But so we talked about scabies. Scabies would not be a linear pattern at the waist. It would tend to be between the fingers.
We did have a scabies
and it wouldn't
case
really be.
also.
Yeah, we did have
Plus-
scabies, and it wouldn't be bites as much as burrows, right?
And then lastly, it doesn't exhibit as between the fingers type of expression. It's a rash that you get to the secretions of the scabies, which don't appear in the fingers at all. It appears in the mid-region
Scabies is out.
of the trunk.
What about pigeon mites?
Yeah. Do you want
Dermatitis.
What are your thoughts on pigeon mites, Dickson?
Well, my thoughts are limited because I don't know a lot about them. But I do know that they're common, and they're so common that they should be causing more dermatologic conditions than they actually do. So the thing is that they do prefer pigeons over people, and therefore if the pigeons are available and they're landing on the windowsills, the pigeons are probably the object of their affection, as it were.
I mean, we do see pigeon mite problems in New York City. And as you're saying, they prefer the pigeons. So it requires sort of a repeated exposure
It does.
to pigeons. And people will find all kinds of other things and bring them in. But you sort of want to get that. And that'll be more in the city where there's many more pigeons. You start getting out into the suburban areas and not as many pigeons.
That's true.
Very, very few pigeons. And, we'll say thanks to the house cats, maybe. I don't know. They're eating the songbirds on the pigeons. But no, so you will see that in New York City. I don't think we have a good history for that. And then I think we should talk about, we'll sort of lump in here with the fleas and what are the pattern of bites, right? I mean, I think we had people mention that your fleas are going to bite kind of indiscriminately all over the place.
Correct.
And they're going to be in sort of this cluster. You're not going to get a linear 3, or 4.
No.
It's kind of classic to see this 1, 2, 3, and then maybe the 4th in a line.
Yep.
And again, more on the central warm trunk.
Yeah.
Because they really are there. It's not like they bite. They bite and then they bite and then so they're actually spending a little bit of time traveling to the warmer, more central parts of the body. So upper thighs, abdomen, thorax.
Yep.
In the bedbugs versus your fleas will be ankles a lot of times, right? They sort of when they get access, boom, they bite.
Right.
So our history is, I think, pushing us in one direction versus another.
A very good direction. I
Found an interesting article, by the way. entitled "Pigeon Mites Will Feed on Sleeping Humans," and it's a woman whose mother, a guy whose mother, 80-year-old mother living in a high-rise in Manhattan, had bite marks on her legs and chest, but no bed bugs were found. It turned out to be pigeon mites.
Yeah.
And if they nest nearby, they can bite you. I didn't know this. Interesting.
Yeah, we have some pretty sophisticated listeners,
Very
right? So that is something that is reasonable to have in the differential, and it's sort of. So what do? We do the Dr. House approach, and we actually we say, you know what, this is pretty concerning. I think you need to call somebody. They call somebody, and then I have a representative picture for these guys to look at, but they actually identify several of these.
Where?
So they call, I hear this story and I say, this sounds for all the world like bed bugs. Mm-hmm. And so they call and they have someone come in, and this person strips the mattresses, and in the seams of the mattresses, they find several rather small, about 0.5 centimeter in length and smaller, bugs, and they actually are bed bugs. So they're able to recover the different bed bugs.
They're pretty big.
They're pretty big, and they're fast. They're not slow. They're fast.
In the seams of the mattress.
In the seams of the the mattress.
In fact,
so should they throw out their mattress?
Well, they bring this to me. I get to see this in a plastic Ziploc bag.
Yeah.
And it's hard. I'm doing the thing with the iPhone where you can magnify it to see them better, to make it out. But and then they actually, and so then they find out like what it's going to cost to, clear the rooms. And there's several different ways to clear the rooms, right? So this is the nice difference between these maybe, and we'll say scabies. The scabies are living on you, right, and burrowing in. These are living somewhere else, and they may take several days or even a week or two between when they come out to feed.
Whose bed did they find it in?
So they find it on the oldest daughter. So that was the first daughter, so it was the other daughter. They find it in the seam there. And they find several, and they put them in a plastic bag. I get to see, but they're told by the exterminators that, for some massive fee, they will come back and they will treat everything. They'll they'll seal the mattresses in plastic.
Not to give an ad plug to somebody, but there is an ad on television that uses a dog that's trained to smell the excrement of the bed bug.
And then what do you do?
Then you have it. So you have choices, and actually this is important because if you can afford it, they come in and they're actually going to spray all these different areas. And the big thing is to seal the mattresses, to get all the clothes, all the clutter, everything out of there. Put it in a very hot dryer for 90 minutes at above a certain temperature, and the exterminators are going to walk you through all this.
The mattress is sealed to kill the bug.
Well, so that they can't get into these seams where they want to live.
So you leave. The seal.
You leave the seal on. So
doesn't that crinkle while you move around? No way. I'm not happy.
Vincent's not doing.
Not happening.
Now,
there are other approaches.
So one is a heat treatment. They actually bring these heaters in. And again, as I was mentioning, 90 minutes above a certain temperature, 155 is what they'll usually do. They keep the entire room hot for that long. And they come in their suits and they heat everything up.
Seal all the windows and doors, and then
they heat the room.
Why didn't the husband get bitten?
I think it was the issue. Well, so there's two, we don't know for certain.
He might have
just been a non-reactor. Okay. The husband was the only family member that grew up in New York City. So he may have
blocking antibodies.
Yeah, he may have become tolerant to bed bugs. The other was, the mom's more involved with the kids, reading to them in their bed at night, sometimes falling asleep in there. So she may have, she may have actually been. And
this happened suddenly. It wasn't always present. So what was the origin of the bed bugs? Do we understand?
So we don't know. We asked a lot of the questions, I think last time. Had one of the daughters, particularly the youngest one, been at any sleepovers recently? And yes, she had been. So that's potentially get a whole bunch of little girls together, right. And then and you think, but nobody, did anyone in school mention this? People don't mention this. You don't think this mom is going to call or tell any of her friends?
Hey,
I just want to let you know we have bed bugs in case that information is helpful. Because we associate this with being unclean. It's embarrassing, and so this
not true, but that's the
psychological.
It's the old association.
Yeah, we used to, when the kids were really young, they used to bring home head lice from school.
Sure.
Oh, all the
and
time.
then you had
yeah.
to do all this stuff. What a pain. I'm glad they...
So there's another approach to getting rid of the bed bugs that you didn't mention, but you will maybe?
Well, why don't you jump in?
It's the use of CO2.
Okay.
So you seal the room off and just fill it up with CO2.
How would you do that, Dickson?
Well, there's a lot of ways of doing it. You know, there
Plastic
are cylinders of CO2.
How
that you
about
can
dry ice?
pump it in there. They have a wand that they could decontaminate this office
A
with.
fish called wand?
No. No. This is a wand called wand. That it secretes a concentrated stream of what looks like particulate dry ice. And you go on the ad for this, and they can decontaminate a computer, they can decontaminate office furniture and phones and things like that using this. It lowers the temperature and essentially freezes the bugs, and then it thaws back out again, and everything is back to normal.
I like the suggestion of burning the house to the ground.
Burning the house to the ground works
Of course,
wonderfully.
if you live in a 30-story high-rise in New York.
You have to move somewhere else.
So you can bring these home from hotels, is that correct?
Oh, absolutely.
Yeah, so there's a lot of
On your luggage.
So there's a lot of advice about that. And so people go to a hotel, and you've ever noticed there's this thing that opens up, and you're supposed to put your suitcase on there. You think, oh, that's so nice, so it's not on the floor; it's up on this raised thing.
Yes.
That actually will help protect you from the bed bugs by
That's true.
raising your luggage up like that.
The bugs won't get into
The
your
bugs
luggage?
won't get
Never
into-
put it on the bed itself.
Yeah, you don't put it on the
Never
bed.
put it on the bed.
Yeah,
Oh,
you
I
don't-
do that all the time.
We all
Yeah,
do.
that's what
We all
everyone does.
do it.
They throw the suitcase, they spread it open, all the stuff
Right.
comes out onto the bed. That's not.
Yeah,
what
I
you
put
wanna
my
do.
backpack on the bed. Usually that's what I can.
Yeah, no, you open
I hear that.
that thing, put your suitcase, and particularly when you're coming to, people talk about, oh, I'm going to Africa. What should I do? Well, if you're coming to New York City, you got to take bed bug precautions.
Wow.
So put your
So where
put
in
your
the
bag
U.S.
on one of those.
Where in the U.S. would I find a hotel with bed bugs?
Everywhere.
Everywhere.
A lot of major cities. We had to mention that San Diego, but New York City, we're
finest
famous.
But
we're
you're gonna
famous for it.
crawl into bed and they're gonna get on you. Do you carry them around, or is it just in your luggage that the bed bugs?
So
go?
they don't like to travel on you.
Okay.
They like to travel on the, they
Luggage.
get into the seams, and that's where they're going to,
Yeah.
and they'll stay after a blood meal. They may stay for a week or two before they go searching for another blood meal.
Right.
So that's also, someone will have bed bugs. And you tell them, oh, I think it's bed bugs. You recommend this. They get the huge bill from the exterminator. And then they're like, I'm not going to do that. You guys go away. And then a week or two go by, everything's all better. Boom. Then they appear again. And so, they may run into the issue of, the denial until it keeps happening because they're not feeding every single night.
Is it a problem to have bed bugs bite you all the time?
As our writers put in,
Question. That's a great question. because one of our illustrious graduates, Baruch Blumberg, who you know of,
Yeah.
and maybe have met, I had the privilege of knowing him, spent a lot of his middle research years in West Africa trying to show that bed bugs transmit diseases, namely hepatitis, which was his big number.
Right.
And try as he might, no pun intended, he could not demonstrate a vector role for bed bugs, no matter what. So a vector needs to suck it in and then spit it back out at some time later on. And apparently bed bugs just spit it out, but they don't suck first and then spit out. They spit out their energic compounds, they suck in the blood meal, and they leave, and that's the end of that. And
That's interesting.
nothing grows inside
I'm
of
surprised.
them. Apparently. Yeah, me
I'm very
too.
surprised because
Yep.
Viruses, as you
Agree.
know, take advantage,
They
even
do.
though they can't take anything.
Right.
They do take advantage of all hosts.
This
And
is
I'll
true.
bet bed bugs have viruses in them, but
No doubt about it.
they're just not transmitted.
Maybe they're
But
not
let's think
human
about-
infectious viruses.
Let's think about the lifestyle. So when a bed bug bites you, is it taking a blood meal?
Yes. That's how it feeds.
All right, and then it goes off and digests the
Yeah,
blood,
That's right.
right? And then if there were viruses in the blood, the viruses would remain in the bed bug, I presume. And then when it bites someone else, does it inject saliva like mosquitoes do? Yeah.
Sure
So
does.
the's a possibility for vectoring.
That's
of
why
virus.
Baruch stayed with it for so long.
Yeah, okay. But they
Yeah.
don't seem to be.
Could not demonstrate a given role for bed bugs in viral transmission.
Did these people clear their house of bed bugs?
So they bit the bullet, they paid the exterminating bill, and they have since had no more bites.
Happy ever after.
Right.
And apparently they're sleeping on the crinkly plastic. I don't know. Do they make covers that? I don't know. Do they make covers that are not crinkly?
I bet. I
right, 'cause
suppose you
you
could find.
got to keep them on, otherwise it's going to be a problem.
So most plastic is pretty crinkly, and I would not stand for that. I cannot sleep on
I think
crinkly
what I would
plastic.
do is I would CO2 under the crinkly material for 2 days to kill all the bed bugs in the mattress, and then I would take it off.
I'd buy a new mattress.
Yeah.
You see a lot of them on the streets because of that.
Yeah, don't. If you find a discarded mattress in the streets of New York
Leave it.
City,
Oh my
do not
gosh.
bring that into your home.
Don't even go near it.
But again, as we're mentioning, if you let's say a bed bugs, you're like, I don't want to spend all that money. There's no health issue.
Correct.
It's just a little annoying, and over time you'll probably become allergic, like the husband seems to have become.
But it's a little bit.
Well, then you always feel awkward when people come and visit you, and
No, but you also feel like every time I go to sleep, I'm going to have these creepy crawlies all over my body. And a lot of people don't like that idea.
yeah. So people often want to get rid of them.
They do.
All right. This episode is sponsored by CuriosityStream, the world's first ad-free nonfiction streaming service founded by John Hendricks, the founder of Discovery Communications. It's available in 196 countries on many platforms like on the web, Roku, Android, iOS, Chromecast, Amazon Fire, Kindle, and Apple TV. What do they have? A wide variety of science and technology, nature, history, many more topics, all nonfiction. Over 50 hours of 4K content, one of the largest 4K libraries on the net. They also have interviews, lectures, maybe even with Dickson Despommier. Stuff like Stephen Hawking's Universe, Next World featuring Michio Kaku, who talks about technology, AI, and other big tech questions. Viruses, a couple of shows on viruses. I haven't checked parasites. I should check. Life on Us, a series that explores the biodiversity of our bodies, including ectoparasites, Dickson. With CuriosityStream,
Thank you, Vincent.
you get real science shows, not just reality TV, monthly and annual plans starting at just $2.99 a month, less than a cup of coffee, or the cost of one title on competing on-demand platforms. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series, completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of Microbe TV and TWiP and all the other shows.
Somebody drilling, right?
Someone's doing dental work.
So I was trying to figure out. So why did you point to Dickson when you mentioned ectoparasites? I'm trying to figure out what the connection is there.
I
But
can go like this.
Talk about
He can't.
ectoparasite. What is the story? They have to drill while we're doing TWiP. Sorry, folks.
okay. Well, so are the mics directional, or is everybody going to get to listen to them doing the brickwork?
They're pretty good cutoffs, but it is loud. All right, we have a paper, which Dickson didn't want to do, but we're doing it.
Not true.
You want to fight?
Not now. I thought we did that.
I'm just kidding. I'm just kidding.
I think it's just that there were so many competing papers this time.
There were, but I'm fine. I said I'm fine. I'm okay.
That's okay.
I read it. I liked it. It's a good paper.
We'll do your paper next week.
No, this
It's
is
not even my paper. I just selected it. That's all.
you
by
know
our
this is a good
friend.
paper, and we actually cite it in our updated Toxoplasma chapter.
Ha!
Can you imagine that? So we are up to date, and that was completely independent of deciding to read this on TWiP.
It's a paper from MBio.
It is.
It's called A Novel Secreted Protein, MYR1, is central to Toxoplasma's manipulation of host cells. The first author is Magdalena Franco, and the last author, who would be the PI, typically is John Boothroyd from Stanford University, the Department of Microbiology and Immunology, which is where I almost went instead of here, and I should have gone there. I should have gone there. Although
You can check which TWiP was John on, by the way.
Yeah, I don't know. It was not too long ago, but he was right here in our office,
Yeah, he was.
right?
Sure he was.
Nice fella.
He's a nice guy.
Toxoplasma. Now, this is an intracellular parasite,
It is.
right, Dickson?
Yes,
And
Vincent.
it has to regulate the host when it's in the cell. So the idea is that it produces things that come out of the cell. What do you call the cell that's inside of the eukaryotic cell?
It's a parasitophorus vacuole.
It's in a parasitophorus vacuole. And what
It's not
we
an
say
phagolysosome.
are these tachyzoites that infect the cell,
They are.
and they can get into a lot of different cells, right?
Look at you.
Is it true that
Tachyzoid.
they
I
can
like
get into a lot of different cells?
You can get into every cell.
And they secrete what we call effectors.
Okay.
With an E, effectors.
Okay.
And one of them is the Rop-tree protein. Remember Rop-tree? We used to call them rope trees.
It's true.
I think you still call them rope trees, don't you, Dickson?
I do.
Over lunch, the
I
rope
enjoy
tree.
the old swimming hole image of that.
Swinging in on a rope tree.
That's right.
Yes. Yes, that would be nice. We should do a TWiP on a rope tree sometime.
I have a nice screensaver picture of a rope tree if you want to see
do?
it. Yeah.
So anyway, the issue in this paper is that it's known that Toxoplasma induces a cellular protein called c-Myc.
Right.
c-Myc was originally discovered as an oncogene picked up by a retrovirus many years ago.
Look at that.
So there's a cellular protein.
Was that a Fred Alt discovery?
He might have.
I think it
Might
was.
have discovered c-Myc. But c-Myc is an important regulator of many, many cell processes. In fact, when you overproduce it, it transforms the cell, so it's involved in growth control. So apparently, Toxoplasma likes to turn up c-Myc. It probably gets some benefit from it. And the question here is, how does it do that?
Right.
That is the underlying question.
Indeed.
In this. What is the effector that upregulates c-Myc? So they do a genetic screen to identify it.
Right. Remember, by the way, John Boothroyd is a trained virologist that moved
he?
into eukaryotic biology. So he thinks like a virologist.
He knows his stuff.
He certainly does. He's one of the most widely respected toxoplasmologists in the world.
So what they're going to do here is they're going to mutate the toxo genome, and toxo is haploid.
Indeed.
So that makes it easier, right?
It makes it a lot easier.
Because you hit a gene, and that's the phenotype, whereas if you had two copies of it, it would be harder, right?
They have them both.
Or you'd have to have a dominant acting, whatever. It's just a lot harder. So they designed a screen to do this. And to do this, they produce a form of MYC linked to green fluorescent protein.
Very
So they can see when it's turned on in cells, right?
Right.
So basically, they
just turned it
they
on.
are. They just turned on c-Myc. So when you take a eukaryotic cell with this green c-Myc and you put toxoplasma in it, they turn green because toxo is turning on the green. It's a lovely screen, and in fact, you can use flow cytometry to look at the population that are turning green in the presence of toxo. Right, Dickson? Do you like that screen?
I love it. Yeah, it's visual. It's a nice visual screen.
A visual screen.
Yep.
The visual display of data.
Do you know how many different fluorescent probes there are
There are
that
a
you
lot.
can use at one time to visualize a cell? One cell.
You're only limited by the machine that you have purchased. How many channels? How many? 12? 16?
I just read an article that said you can do 1,400 different
In
fluorescent
one flow
probes.
cytometer?
In one cell, you can do 1,400
I don't know if
different.
I'm okay with that.
I'm not okay with that
think-
at
Well, this
all.
was a science publication.
Oh, the
so-
hell with science. I think they stink and they're wrong.
Now, I think you can...
No, it was a science. It wasn't the Science magazine I was referring to. I think it was.
I think more like 20.
I think
is
if
it,
you
right?
so if you tag with rare earth metals, you can use mass spec and you can do analysis getting over 100. But if you're using fluorescence and the whole issue with overlap,
It's a spectrum.
really about 30 is I think the max that
But
people
a lot
are.
of them locate to different parts of the cell, so you can get around the overlap in some ways by looking at that.
I'm skeptical. Can I be
No,
skeptical?
of course you can. I was too. I mean, I didn't think that was up to that number, but that's what they said.
Well,
and
they're
actually
certainly,
here they're using two colors, right?
yeah,
They've
Just
actually
red and green. That's
They-
it.
they
Yes,
also
they've
label
labeled
it.
toxo red,
Right.
and the induction's green. So you could see. Let's look at all the ones that got infected.
Yep.
And let's see. Because you could have ones. Oh, they didn't turn green. Well, they could not turn green because they weren't infected.
That's right.
So you want the red marker they got infected, the green marker that they're upregulating c-Myc, and what they're really interested is the red ones that aren't turning green because they're going to then
Right.
compare those to the red ones that do turn things green.
Right.
So they mutagenize toxo, and then they use this assay to look for, as Daniel said, red cells that are not green. And they have to go through multiple rounds and selecting and remutagenizing. But eventually they get a bunch of candidates that don't make green. In other words, they don't turn on MYC when toxo infects them. And then they just do whole genome sequencing on each of these.
Whole genomes.
At one time you couldn't do this.
It's amazing.
But they can just do the whole
That's
genome of
amazing.
each of these mutants and find out where the changes are.
So everything else is the same except this one little part, and bam, there it is.
Isn't
that's
that
amazing.
Isn't that remarkable?
That's just amazing.
that we have. Do you know the size of the toxogenome offhand? Let's look it up because I think this is important.
No, I don't know. Sorry.
Say size of Toxoplasma genome. You think we'll get a hit, Dickson?
I know we'll get a hit.
You're going to get criticized. We should know that. We should have it memorized.
It's only 80 megabytes.
Is that all?
No, sorry, megabases, not megabytes.
Megabytes?
Megabases.
I thought it was 8.1. Am I?
Oh, well, it says here.
I'm serious, actually.
80 megabytes, but that's nothing.
They're rounding. They're rounding. I think they're rounding.
This is tiny because, human genome is 8 billion. Is it 7 billion bases, the human genome? Size of human genome. Dickson, why don't we?
Genes.
No. 3 billion base pairs.
3 billion.
So 80 megabytes, 80 megabases, 80 million bases is relatively easy to sequence. So that's why they could do all these mutants.
But they already have the parent sequenced. They just have to find the differences.
Well, you still have to sequence the whole thing.
I see.
It's not like crystallography.
I see.
Or
subtractive hybridization.
Yeah.
So they end up, I don't want to go through all this. They have a number of different candidates, but one protein ends up being, the gene for it ends up being mutated over and over again on multiple isolates, and they call it the gene is actually called TGGT 1_254470, but they call it MYR1 because that's, what is it?
It's
essential for
MYC
regulator number 1. Yes, much easier than TGGT1
Just flows out of your
underscore Dickson. You could be underscore. You could be underscore Dickson.
I could.
So I'm
certainly underserved.
What is MYR1? They, of course, would like to see where.
What is it?
Well, it's abundantly expressed through the major infectious stages, including tachyzoites, bradyzoites, and sporozoites. But it is not produced in the intraepithelial stages in the catgut. So they think it's not key to the sexual stages of the parasite.
Right.
All right. So it's highly conserved. It has transmembrane domain, so it may be a membrane protein. They delete it from the genome and show that you need it to have induction of MYC. And then when they put it back, it complements the deletion. So you want to do that, right? Just because you found this in a strain, you want to actually delete the gene from toxo and then say toxo can no longer induce MYC.
And it's important to add it back to show that that's the only difference.
And the growth rate of the parasite was noticeably different.
When you delete this thing. It appears to be important, yes.
However, it didn't kill him.
Did not kill him. No, it did not kill him.
Redundancy of nature. I'll bet you it's got another gene that probably compensates for it.
It might. Nature is redundant.
Because it's not.
Nature is redundant.
This is not an essential for the growth of this parasite. It's an enhancer.
I think if you threw these parasites in nature, they wouldn't survive. They grow too poorly. Right.
This is the world's best parasite. Remember that. This is the better than any other.
Are you infected?
I haven't tested myself, to be honest.
Am I?
I don't know.
Are you, Daniel?
Probably.
Because you've been to Cambodia. You've been to Cambodia.
Steak tartare. Yeah, you do a lot of travel.
I do a lot of travel, and maybe when I was younger, I was more risky in my dietary adventures.
Right. I mean, the other parasite that comes even close to this for humans is pinworm. And this parasite is found in all animals, in all tissues of all animals. I don't know even a virus infection that does that, Vincent. I'm sorry to say this, but I don't know one that does that. Do you?
Yeah, I do.
Which one?
The nanoviruses infect every human on the planet, as do retroviruses.
And all the animals, too?
Yeah.
And all the cells of all the animals?
Yeah. Okay. Every human on this planet has in every cell of his or her body in its genome a retrovirus. They're the most fabulously successful
viruses those are not,
ever.
They don't pop out every now and then.
You want to make qualifications
This
here?
thing does. this thing causes disease.
Dickson, this protein is cleaved.
It is into two.
But they don't know what that means.
So it's a dimer, maybe.
No, it's just cleaved.
Yeah, but they say both parts are active.
Both parts are active.
That's what they say.
But they don't know why it's cleaved. Okay.
So do they have the gene for it?
Well, yeah, they do have a gene.
And then what is it? How much similarity is there between that and anything else?
I think it's pretty unique. They don't actually.
I mean, they hemmed and hawed a little in the discussion.
They did, because I would like to see a structural paper on that.
They don't know. It's a novel protein.
I understood.
They saw homology with the Pseudomonas brasilianum protein. I mean, they were trying to find, but no, I think you're right, Vincent. It's unique. They did not find a very high homology.
They make antibodies to it, and then they stain, and they find it in the parasitophorus vacuole.
There you go.
And what do you think about that? What is this parasitophorus vacuole?
So you have to. I will tell you. I can distinguish two things for you that will set the stage for.
That's it. In your whole life, you can just distinguish two things.
Well, you only have two things. You either have a phagolysosome,
right,
or you have a parasitophorus vacuole. A phagolysosome is designed to digest stuff. When this parasite enters a host cell, it initiates all the stages necessary to prevent the formation of the phagolysosome, because it shuts off the hydrogen pump. It does not allow fusion of lysosomes to the membranes. It does a whole bunch of things to subvert the host cell into allowing it to grow in the face of very, very, very strong protective mechanisms at a cellular level, which prevents the cell from being infected to begin with.
Is something in the parasitophorus vacuole, is that on its way to being injected into the host cell?
Well, or facilitates other things, as this case is to go into the cell.
Daniel, they don't look in...
So it's like a shuttle.
They don't actually look in an infected cell and stain to see if this makes it into the cell, right? Although it must, because it has big effects on c-Myc, for example.
Yeah, I mean, they're actually talking how it may affect transport from the parasitophorus vacuole into the cytosol.
Correct. So you mentioned to begin with rhoptries.
Rhoptries, yeah.
And granules within the rhoptries. Right. And micronemes, those are another group of organelles
Wonderful
in the front of the organelles.
Wonderful
I love the words. They're great words, right? And they secrete things. And so there were two of them that they discussed, right?
Yeah.
GRA, what was it, 17 and GRA 24?
GRA 7 and 24, yeah.
And the 24 apparently is linked to the process of this protein getting it out into the cytoplasm and into the nucleus of the host.
Yes, to alter host transcription, it needs to go in the nucleus. So this is in the parasitophorus vacuole. It also so c-Myc messes up when you turn on c-Myc. It messes up the cell, and it alters the cell cycle. In fact, what it does is the DNA becomes 8N instead of 2N, which is diploid. It becomes 8N.
How interesting!
When you infect with these.
So gene duplication at the entire genome level. Is that basically it?
It's duplicating the chromosome without cells dividing.
That's incredible.
It's inhibiting cell division, but the DNA is
incredible.
And this protein MYR1, if you take that away, that doesn't happen anymore.
Do you know what other parasite does this?
So that's called disruption of the cell cycle, and MYR1 is needed for it. What other parasite does it?
Does this. That's right.
Which one?
Begins with T.
Toxoplasma.
That's the one we're talking about.
Treponema.
Trichinella,
the one you used to work on?
The one I used to work on.
Which you could be working on this very day.
I'm working on it all day, all night. I think about it all the time.
If you wouldn't be doing parasites. You wouldn't be doing podcasts, I mean.
Of course I would. Come on, you do viruses and you do TWiP.
I wouldn't have met you if you'd still be running a research lab.
Maybe not.
And I wouldn't have thought to come start a podcast with you. So things in the world are so intertwined.
But we started our podcasting before I actually stopped working in the lab.
All right. What other things are interesting here? They look at a bunch of phenotypes.
I think the fact that the parasite gets something into the host nucleus via an intermediate molecule is fascinating.
So they actually find that this protein is needed for these proteins that you talked about, the GRA. That stands for granule proteins.
Of course.
To get out of the parasitophorus vacuole into the host.
Right.
If you take away MYR1, these proteins can't do that. So it's regulating their transfer.
You know what else goes on to that parasitophorus vacuole is that the mitochondria of the host cell aggregate at the interface.
Oh, yeah?
Which may aid in the energetics of the parasite. We don't know that for sure, but it's a fascinating association.
You know what's interesting? So this MYR may affect GRAs getting in the cell, and they may, in fact, be messing up.
Exactly right. Exactly right.
So if you actually got rid of those, would you then lose your MYC upregulation?
So there's an engineer, that's the molecule, and then there are workers that they send out to do the jobs.
Right. So they also show that GRA24 getting into the host nucleus.
Yep.
That doesn't happen when MYR is absent.
No. And yet, the organism still carries out its
So maybe
life
it
cycle.
helps get it in. Maybe it binds to it and helps get it into the nucleus. Maybe it has a nuclear localization signal.
Yeah.
Right?
Yeah.
And that's about it. They do infect mice.
That's about it.
Dixon, one more thing. Yeah, we have to move here because we don't have a lot of time.
Right.
And we have to hear a case result. They infect mice with these mutants. What do you think happens? They infect C57 black mice, IP, and they look at survival and weight loss. So if the wild-type virus, they're all dead by 9 days.
Virus, you said virus?
Viruses are the only things in the world. Wild-type toxo kills mice within 9 days. What do you think happens with the MYR deletion?
Well, I already read this article, so I know
I
that
just
it
want to
doesn't kill
see
them.
if you remember. It doesn't kill them. They all
Of course I
survive.
remember.
They all survive. So it's necessary for full virulence, which is not surprising because it has such an importance.
So wouldn't this make a great vaccine?
That's a good question. If it grew properly in the host, right? Because if it's feebly growing, you might not have enough of an antigenic boost.
So there's one that they created by temperature sensitivity, right? It grows at a reduced temperature, so they inject it into the skin of an animal.
This is a vaccine
And
candidate.
and it stays there and it replicates, but only in the skin because it's temperature sensitive. Once it reaches the temperature of the core, it dies. That's a clever use of biology. Elmer Pfefferkorn.
This was done in dog. Was this done in dogs?
Cats. It did it in cats.
So the summary, Dickson, is a novel exported protein is necessary for the function of several secreted effectors. Those are the granule proteins. Right. The absence results in attenuation.
Right.
The mechanism of its function is not known, but it's probably involved in transport of effectors across the parasitophorus vacuole, where they can then access other compartments of the host.
So it shuts off things from coming in from the host and facilitates things going out.
I liked it because it's a nice screen. You identify protein. I'm sure they're going to work on this for many years, right? Clever genetic screen.
That's right.
All right.
By a clever biologist.
Let's do a case study.
Okay.
Hit us with a good one here, Daniel.
We have 14 minutes.
All right. I can do it. I have to admit, when I was putting this one together for everybody, I had to check to make sure I hadn't presented it before because it's another fascinating
You've done a
case.
lot of them now.
So here we have an 80-year-old woman living in the Washington Heights portion of Manhattan, presenting to the hospital with 10 days of watery diarrhea. So to give people context who are not from the Washington Heights area, this is a very northern part of Manhattan.
Where we are right now, right?
Where we are right now. This is a local case. You're a little bit south of the George Washington Bridge, right? The one that Christie shuts down periodically.
We're 10 blocks south.
This woman reports no blood in the stool. She says that the diarrhea is continuous, even waking her up at night. Okay. The stool is watery and is not particularly strong in smell.
10 days duration, you said?
10 days of watery diarrhea.
Yeah.
Right.
Not smelly. Okay.
Not smelly.
Is it lipid full of lipid? What do you call that?
So the stool does not float.
Diarrhea.
So she's not reporting floating form stools. And actually what Vincent, what you bring up is often like with giardia, which we've discussed before, when you have a fat malabsorption, you end up with a lot of fat, your stools will smell, will float.
Correct. Because the fats get down into the microbiome and then they get altered.
She doesn't report any fever, but she does report sweats at night sweats. So like waking up at night, just in a drenched sweat, having to change the bed sheets because it's all wet. It's been going on for 10 days. Okay. And she says that in general, she's not feeling well.
She lost weight.
I wouldn't either.
She's losing weight.
She is losing some weight.
Loss of appetite?
Yes. Yes, she has some loss of appetite.
Has she been traveling?
She has not been traveling.
Traveling.
She, just to give the context, I think it will be fair here. She was born in the Dominican Republic in the early 1930s, right? This is an 82-year-old woman. But she moved to the U.S. at age 30. So she moved to the U.S. 50, 60 years ago. And she occasionally goes back and visits the Dominican Republic, but she hasn't been back for several years. When she was in the DR, she was not I always ask, "Are you from the big city?" And she grew up in a rural area, not from the big city.
So interestingly, the people from the DR make up the bulk of the population up here, right?
It's true.
In fact, when we took a cab to the Bronx Zoo, our driver was from the DR. Remember?
So that's Dominican Republic.
Exactly.
Yeah.
So the question I have next is, what time of the year was this?
So this was. Let's see if I jotted that down. This was in the spring.
Spring. Thank you.
Yep.
And she's living alone?
She lives with an extended family.
Anybody else sick?
She's the only one that's sick.
Okay. And what does she eat usually? Does she eat meat? Does she eat cooked food, vegetables?
She eats a whole assortment of cooked foods. She eats a lot of classical Dominican food, a lot of rice and beans and chicken, things like that.
Drink tap water or bottled water?
She drinks regular tap water.
Which here in New York City is wonderful, you know.
Best tap water in the world.
It's called dilute soup.
Dilute soup. And she's not traveled. What does she do? She stays home most of the day?
She's not working. She stays home.
Any pets?
No pets.
No pets.
No.
Any neighbors with pets?
There are dogs. Fair number of dogs.
Cat sit or dog sit?
She doesn't. Yeah.
Does she sit outside on the front stoop most of the day, or what? She stay inside?
So she stays mostly inside. I should give you a little more past medical history. She has a history of reflux, high blood pressure. She has had some cognitive decline. Mm-hmm. She's not quite as sharp as she was before. And a couple of months prior to this, she was diagnosed with temporal arteritis. Giant cell arteritis involving the temporal arteries was biopsy proven.
That sounds pretty serious.
Yeah. Well, they put her on some prednisone, and she's doing much better.
She's on blood pressure medicine?
She is on hydrochlorothiazide. She's on a proton pump inhibitor, and she's still on the prednisone for the temporal arteritis.
Okay.
She had her gallbladder removed. It's her only surgery. Not allergic to anything. She doesn't give us any family history of any problems.
Does she have HIV?
She does not.
Any of her family, this extended family she lives with, what do? They go out and work in the world, right? Mm-hmm. Any particular interesting things they do that would bring something?
All
home?
Working in the city. No one with any particularly interesting occupations. Working in stores, things like that.
Any taxi drivers?
No taxi drivers, interesting enough.
Any of them take trips that they may have come back from the Dominican Republic?
Yeah.
No. Have they been to the zoo?
Not that they report. No.
They keep
more people should go to the zoo.
Is this woman married, or is her husband deceased?
Her husband has passed away. Has died.
All right.
She's 82. Men, we don't usually live quite that long.
Has she ever had this condition before?
Diarrhea.
Watery stool.
This is unique.
According to her diminished memory.
She's had loose stools. She's had diarrhea in the past, but this is really bad.
This is different.
Is this accompanied with any discomfort, abdominal discomfort?
Yes. Her belly is uncomfortable. Yeah.
Does she eat fresh fruit?
Is it always uncomfortable?
No, it's new. She was, she said she was in reasonably good health before this, but now she feels horrible.
How many stools a day does she have?
It's almost, she says, almost continuous.
Well, she must be drinking a lot of water, right, to make up for the...
She is.
And her belly is always uncomfortable, or just in bouts? I'm sorry, Dixon, I'll stop.
Right?
Now.
No. It's.
I won't forget my question.
No, it's been uncomfortable now for over a week.
Okay.
Does she eat fresh fruits?
She does.
Like, for instance, not peeled fruit, but other kinds of fruit, like berries.
I don't know specifically about berries, but she does report fruit. Berries? We did not ask specifically about berries.
Okay.
Are you going to give us any physical exam or labs?
Yes. So her temperature is 38.1, blood pressure 116 over 78, heart rate
116?
116 over 78.
Got it.
Heart rate is 105. Mm-hmm. She's breathing comfortably at about 12 times per minute. And I'm going to give you the exam, and I'm going to give you some labs. Mm-hmm. So on exam, she looks ill. She looks like she's not doing well. And she has a diffusely tender abdomen with, interesting enough, decreased bowel sounds. Her liver and spleen are not enlarged. She knows where she is. She knows her name. She's oriented. And she has a rash on her abdomen. And I'll describe the rash because maybe that's helpful. It's sort of this odd patchy distribution, and it looks like multiple thumbprints. And it's only on the front of the belly, and it seems to be extending out from the umbilicus. So it's like a darker red thumbprinty rash sort of extending out from the umbilicus. And I will give you guys some labs as well.
She has a diffusely tender abdomen. That's it.
Diffusely tender abdomen with this rash.
Got it.
The watery stools. And I should help people. Like, so I mentioned the fact that the diarrhea is continuous even when she wakes up. And we make this distinction. Sometimes they have like a malabsorption, or you eat something and out it comes. Mm-hmm. But the continuous, we usually think of as a inflammatory secretory diarrhea. So the stuff is coming out of the intestines even while she's sleeping and not eating anything. She's drinking, fortunately enough, to keep up with this fluid loss. But the fluid loss seems to be a secretion by the intestines. Mm-hmm. Her white count is 8, so it's still within the normal range. 78% polys, 14 lymphs, 4 monos, 1 eosinophil. So she's got 1 eosinophil. Mm-hmm. It's all lonely. Her sodium is a little bit low at 129.
It should be.
Her BUN is 15, which is not particularly elevated. Creatinine is normal at 0.6. And I'm going to give you a couple things. She's HIV negative. A urine histoplasma test is sent, which is negative. Right. She has a quantiferon gold test, which is done. That's a TB blood test. That's negative.
Quantiferon, you said?
Called quantiferon gold. Maybe we'll talk about that. It's sort of
gold.
It
sounds like a tequila.
It's like a PPD in a bottle, right?
Like
instead of injecting the purified proteins under the skin and seeing if your body reacts, you take the cells out and ex vivo you test them for reactivity.
I see.
It's a great test if done correctly. It's technically challenging. I seem to have experienced at a lot of these facilities. All right. And so I'm going to leave everybody with, you guys can ask a few more questions, but, what are people thinking? What tests do they want?
I know what I would want.
Okay, what would you want, Dickson?
I want a ova and parasite stool examination.
Okay, so you want ova and parasites. So we're going to send stool for ova and parasites.
That's right. I want that one.
Okay. So we'll send that off.
Thank you.
What else would you like?
No.
Nope. So far, that's it. That's
it. You're good. That'll do it for you.
I would do the other multiple tests for parasites as well. There is an immunological test that you can use to detect antigens for a variety of parasites, and so I would do that one as well. Wouldn't omit doing microscopy, but I would add to it. How's that?
Okay.
I would like to wrap this up because I have another podcast in 5 minutes.
Right.
All right. So we'll wait for some emails and
will.
You can find TWiP at iTunes, microbe.tv/TWiP. Also, Google Play Music. Consider becoming a patron of TWiP and the Microbe TV family of science shows. You can contribute as little as $1 a month at patreon.com/microbe TV. And of course, do send us your questions, comments, and guesses about our 80-year-old woman with diarrhea to 83. How old is she?
I thought she was 82.
82? I had written down
82. Nobody was paying attention.
82. Okay. TWiP at microbe.
Soon to be 83.
TWiP at microbe.tv.
Sick.
Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Oh, pleasure as always.
Dickson Despommier is at Racaniello.org. Thank you, Dickson.
Thank you.
And I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.